Thanks everybody. It's a pleasure to be here. I appreciate everybody attending both in person and online. My name is John Bashkin. I'm a co-founder and the Chief Strategy Officer at S2 Genomics. My background originally as a scientist, degrees in chemistry and physics, postdocs in biophysics, and then I've been in the life science tools business since the early 1990s. And I'll take you through a little bit of the history of our founding team in a moment. S2 Genomics is a life science tools company. We are focused specifically on automating sample preparation for specific areas of biology research. Particularly, we take as an input pieces of solid tissue. This could be a biopsy from a tumor, a piece of lung tissue from a mouse model, microdissected organs from a honeybee. And we take that solid tissue and we very gently dissociate it into all of the different types of cells or cellular nuclei that go into making up that piece of tissue. This is a critical first step for a wide array of downstream technologies and assays that all work at analyzing biology at the single cell level. These applications dominate modern biology research, both basic research and clinical research, and will soon be entering clinical applications in diagnostics and in manufacturing for cell therapies. The company was founded in 2016. We're located in Livermore, California. And the three principals, myself, our COO, Bruce Leese, and our CEO, Stevan Jovanovic, are all very experienced. We've been doing this, each of us, for 30 years or more. We all first worked together back in the 1990s at a company called Molecular Dynamics. These are some of the systems that we developed over the years, either as a team or in various guises at different companies, starting with this instrument here, the Megabase 1000 in the upper left. I was the scientific lead on this product. It helped launch the modern age of genomics and was responsible for about $2 billion of revenue from instrumentation and reagent sales. It really helped to launch genomics in the 1990s and early 2000s. We are a product revenue stage company. Our first product, the Singulator 100, was launched right at the end of 2020. We shipped our first 3 systems in December of 2020. And despite doing that in the midst of the pandemic, we now have 80 installations around the world. The instrument itself sells for $37,500 in the US, and it's a standard razor blade model that you have in life sciences. We have standardized single-use disposable cartridges, along with reagents that create a revenue stream for each instrument placement. All of the development of this system, the ideation, the R&D, the mechanical, electrical, and software engineering, the setting up of the manufacturing supply chain, the manufacturing itself, all of the commercialization efforts, sales, setting up and managing our distribution channels outside the US, all of that was done with a group of just 6 people. And we were able to do that because we have all done this before, and we're all able to, wear multiple hats and be very efficient. In addition, the 3 principals, we took no salary in the company until 2021. So we have been very capital efficient. We are all heavily invested in the company. We have a lot of skin in the game in being committed to this company and making it successful. We sell our systems into large research hospitals, genomics core facilities, comprehensive cancer centers, nonprofit research institutes, as well as biotech and pharma companies. This is some of the feedback that we've gotten from customers. After a demo at a university, the graduate student who was running the demo admitted to me that she started to tear up when she saw the results, because what we are doing is replacing manual protocols. These protocols can take anywhere from 2 to 6 hours. They're very hard to do reproducibly. They often fail. And we're enabling somebody to walk up with absolutely no training and execute these protocols, in most cases, in about 5 or 6 minutes. Another Genome Core director told us that, quote, the early users at the Institute have all given him the same response. They're going to be telling everyone in their lab about the singulator. And from a poster that was presented earlier this year from the Stowers Cancer Center in the Midwest, the consistent results from the singulator have allowed labs to reduce isolation time, leading to the design of additional experiments that were not possible with the manual methods. And I think this captures a really key thing about our systems is that although they're convenient, they're faster, that makes them a really nice to have product in a lab. For many researchers, they're in fact a need to have because they simply can't do their work without a system like the singulator. So why do they like the system so much? It's incredibly easy to use. It's very easy to develop protocols for your particular tissue. And these are the kinds of results that we enable them to get. These are data from a tumor, from an adenocarcinoma, a lung cancer patient. And what we did was take that lung tumor, isolate all of the cells from that tumor, and then do RNA sequencing on them using single cell genomics. And that data then tells you all of the different cell types that are present in your sample. And if you were to do this by hand, you would largely get out the immune cells, the B cells and the T cells, because those immune cells dissociate quite easily. But what you're going to have a really hard time getting are the cancer cells themselves. And what you see in this plot here, every cluster that you see, every different color, corresponds to a different cell type. And this orange cluster over here, those are the cancer cells. And you're really just not going to see those very well if you do this by hand. But if you use the singulator, you get a very nice balanced cell type representation across all of these different cell types. In another example, this is from another lung cancer patient, squamous cell carcinoma. And again, these are the cancer cells up here, and you see the immune cells, you see the other types of cells that should be present. And we can look into the genomics of these specific cells and know for sure that those are in fact the cells from the squamous cell and not from neighboring healthy tissue. One of the other reasons customers really like our system is that it's so easy to develop protocols for their particular tissues of interest. These are data from a mouse kidney. And what you see here is that there aren't very many different colors. So this was the first attempt at using a protocol to isolate cells from mouse kidney. And what we found is that you got a few different types of cells, and that's it. All of the other cell types were basically killed off during the isolation process. But with one click of a button in our GUI, in our user interface, we can change that protocol, and now this is what you get. And now all of the different cell types that you expect to be present in that piece of kidney tissue are now there. The immune cells, the endothelial cells, the epithelial cells, the NK killer, the natural killer T cells, neutrophils, all of the different cell types that you would expect are now present. And they're alive, they're viable. You can culture them, you can test drugs against them. This is a prime source for healthy primary cells for doing your assays, whatever they might be. And finally, this is a very new but very, very exciting result. This experiment was taking heart tissue from a mouse and processing it on the singulator and then doing single cell sequencing on that tissue with a company called Parse Biosciences, one of our partners. And the exciting thing here is that we're seeing all of these different types of cells from the heart, including this orange cluster here. These are cardiomyocytes. These are viable cardiomyocytes that we can isolate from heart tissue. This protocol takes about 30 minutes. It's completely automated, and now you can get viable cells out of heart tissue in a very fast, very reliable way, and do your tox assays, do single cell sequencing, whatever you might like to do. It's a very, very exciting result for our customers. I'd like to show you a little video of actually how the system works. This is a video that was not produced by us. It was made by one of our customers to advertise the fact that they had one of our systems in their lab. So hopefully it will play and the audio will play. Again, this was produced by one of our customers, and they sent it to us.
Sometimes when we're discovering genomics, we'll use the Singulator 100 by S2 Genomics. The Singulator 100 is a cartridge-based tissue dissociator that dissociates tissues into single-cell or single-nuclei suspensions. It's so easy a child could use it, and somewhere out there one might. To dissociate solid tissue into a single-cell suspension, a user need only to provide 4 things: the tissue they would like to dissociate, 15 mil conical tubes containing the appropriate buffer and enzyme, and a positive attitude. The instrument software can be used to modify run parameters, for example, selecting the desired temperature or duration of incubation, or the intensity of disruption. Simply add your tissue to the cartridge, put your buffer and enzyme in the correct locations, slide the cartridge into the instrument, and it's party time. There is even a tiny camera to provide you with blindingly high-definition live video while the instrument is running. You can watch and be amazed as your tissue is slowly but inevitably dissociated. When the run is complete, you can remove your sample from the cartridge and proceed to your preferred method of QC. Look at all those cells. Now, you might be saying to yourself, certainly, it can't get any easier than this, but that would make you a liar. Nuclei isolation is even simpler, faster, and in some cases more stylish, with the S2 Genomics formulated reagents already housed on the instrument, and the standard protocols taking only 7 to 12 minutes to complete. In most cases, translating well-established manual protocols into the Singulator 100 run setup will generate expected cell counts and viabilities. In cases where your dissociation protocol may be less optimized, the Singulator 100's fully automated, modifiable workflow allows for precise control over run metrics for optimizing protocols to give the best possible performance. In these instances, like when working with teeny tiny tissue types, sequencing and discovery genomics and the technical support team at S2 Genomics are always willing to offer advice.
Well, I hope that video played okay, and you could hear the narration. It gives you a flavor of how the system works, and also how excited our customers are to be using it, and how much it helps them in their research. We have a nice product pipeline. As I said, the Singulator 100 was launched at the end of 2020. Our second generation product, the Singulator 200, will be launching in a couple of months at the end of this year. And the Singulator 800 is now in concept phase and will be launching at the end of 2023. These systems are used for single cell genomics, for cell biology, even for manufacturing materials for clinical trials for cell therapy purposes. People are isolating cells from tissues that range from human to honeybee, Drosophila, Planaria, rodents, mouse models. All kinds of different tissues are being processed on our systems. Now, these first two products, the 100 and the 200, are very nice products. They've given us a presence in the market. They've established us as leaders in our space. They're generating nice revenue, a nice customer base, and nice reagent pull-through. But the Singulator 800 is really transformative for the company. It increases the throughput of the samples from 1 to 2 to 8, but in addition, it dramatically expands the type of applications that can be done on our system, and dramatically expands the potential customers' install base, and also the opportunities for generating revenue from the reagent pull-through. So we're very excited about the launch of the 800, And I'll talk to you a little bit more about our use of funds towards that product. Everything that we have done to date, the launch of the 100, the complete development of the 200, the initiation of the 800, generating revenue, having almost 100 systems installed now around the world, all of that has been done with an investment of just $10 million. So very, very capital efficient. Today, the company has 21 employees. We've just moved into a brand new 11,000 square foot facility in Livermore, California, where all of the instruments are manufactured, all of our R&D is there. We've also just had issued a very broad patent that covers the fundamentals of our automation methods for isolating nuclei. We have direct sales in the US and a global distribution network that I established and has continued to expand that now represents about 50% of our sales. We have coverage all around Asia Pacific, in Canada, throughout Europe, and now recently expanded into the Middle East with a distributor for Israel. And I said all this has been done with just $10 million of investment.
Did you want to do some Q&A?
Sure. This is just quickly our financial forecast.
Yep.
And the key metric here is that at the end of 2023, we're scheduled to go cash flow positive. And what that means is that should we decide to operate the business accordingly, our current raise, Series A raise, will be the last money that we need to raise. I think that's a key point. And then finally, I'll skip the competition slide in the interest of time. We can talk about that more in the Q&A if you wish. But what we're doing right now is raising a $15 million Series A. And the purpose of this money is to dramatically expand the size of our commercial team so we can capitalize on our momentum and really capture this market, to complete development of the Singulator 800 and begin development of the Singulator platform for clinical applications. And again, we think that we have confidence that this is in fact the last money that we're going to need to raise before turning cash flow positive and hopefully, working towards a successful exit for the company. And with that, I'm happy to take any questions.
Got a question here, yeah.
Are most of the revenues from capital equipment sales, or is there consumable streams later?
So the question is, are there consumables involved with reuse of the product, or is the majority of the revenue come from the original sale?
That's a great question. So there is a reagent pull-through. Right now with the 100, each instrument has a pull-through of about $8,000 a year. That goes up dramatically on both the 100 and the 200 next year when we introduce additional reagents for the system, and continues to go up dramatically on the 800. So with the 200, the reagent revenue pull-through should start to approach the sales cost of the instrument itself on an annual basis. And I can go through the details of that model and what we're doing to execute on that. But it's why the financial model for the company changes so dramatically, first with the 200 and then with the 800.
Sure, John, and we can discuss that at dinner time.
Absolutely.
I just want to go back to your financial forecast.
Sure.
So are the 2021 and 2022, are those actuals?
Yes, so 2021 is actual. 2022, we're on track for that. We're obviously in Q4, so we're very close to hitting that number. The numbers really hurt this year by COVID, particularly from China. We have a lot of customers in China. We were very successful with our 2 distributors there in 2021, and they had big plans for 2022, but the country was just really largely shut down for most of this year. So that really hurt us this year, and we expect that to recover obviously next year.
Right, and then you're showing some expenses that are high in year 1. I don't know that there's no breakdown on that, but I guess that You got some R&D, marketing. So in year one, you had your own distribution channel, and in year two, you also added a distributor. Is that what happened?
That's right. So I set up distributors in 2021. I set them up for Canada and around APAC. And that was largely driven again by COVID. We didn't anticipate doing that, but in 2021, Japan and Korea and Taiwan were all open. They were all functional. And so I pivoted and quickly set up distribution networks for Japan, Korea, and Taiwan, and Australia, New Zealand. And so we were very actively selling internationally in 2021. And then we continued to expand that distribution network through Europe, and the rest of APAC this year. And now I have a full-time distributor manager who is continuing to expand the network around Europe and into the Middle East.
And so your customer are labs. I mean, so who's the payer for this product? Who buys it and who pays for it? Insurance.
So the systems are paid for in core facilities. They're paid for off of the operational and equipment budgets out of those core labs. So at a large research hospital, they'll have a dedicated genomics core facility. They'll need to buy our equipment. Individual researchers will buy our equipment based off of grants. Government agencies like USDA, the NIH have bought our systems. So they buy our systems out of their equipment budgets, and pharma and biotech as well. All of the groups that are doing single cell genomics, primarily in pharma and biotech, are interested in buying our system, and we have a number of placements in both of those sectors. So it's bought off of their equipment budgets. I'm sorry?
How big is that market? I mean, your device, is your device a replacement device, or is it something that's brand new to the marketplace, or you're replacing a competitor product?
Right. So the vast majority of customers in genomics right now are still processing their samples by hand. So we are replacing manual protocols that can be highly variable from person to person and lab to lab. They can take many hours to execute, and for many tissues don't work at all. So that's our primary competition, is evangelizing and converting people from their manual protocols over to an automated solution. We do face.
And how big is that market? How big is that market, John?
So our beachhead application is for single cell genomics. And our addressable market just for single cell genomics is about 500 million. But the ancillary market.
500 million worldwide?
Worldwide, just for our current beachhead market. But once we start to roll in the cell biology applications, manufacturing for cell therapies, all of these markets are really significant and dramatically increase, our SAM, our serviceable available market. We have already sold one system to a biotech company that's using our system to manufacture materials for their clinical trials for cell therapy. So that's not a future, that's starting to happen right now.
One more question?
Yeah.
What's the gross margin on distributor sales versus direct sales?
So the gross margin on distributor versus direct sales?
That's a great question. In the US, our instrument margins now are 60, 65%, something like that. Our reagent margins, it depends on the reagent and the cartridges, but on average they're about 80%. Most of that is driven down by the manufacturing of our injection molded plastic cartridges. Right now, there are some manual processes involved in that. As we scale up, we're increasing the amount of automation. So those margins will go up. We sell to distributors at a 20% discount below our US list price. So our margins go down to was about 50, high 40s on the instrument for distributor sales. The model right now, because we're such a small team, is to continue to work through distributors internationally. But as we continue to grow, we're going to look very seriously at taking that territory back and placing sales managers strategically, primarily in Europe and APAC, and taking some of that territory back and selling direct.
I think we had somebody earlier say that the most likely exit for devices is acquisition. Who's the most likely acquirer of you?
So,
Food
companies like Thermo Fisher, Becton Dickinson, um, uh, Life Technologies, all of the major tool players, QIAGEN, all of the major tool players, companies that have gone through a lot of consolidation. They're all potential acquirers. PerkinElmer, all of these companies are potential acquirers.
Excellent. And you have a time frame for that? 2 or 3 years, 3, 4 years?
Our expectation was that we would hit certain inflection points that would make us an attractive acquisition target. The first inflection point was when we had about 100 systems deployed. That gives us a reproducible, reliable business model around reagent pull-through. The second high value inflection point is when we start to be able to process tissues that have been preserved by FFPE. And that's going to happen in Q1 of next year. And then the third likely inflection point really will be with the launch of the 800 platform, because that's so transformative in terms of our available market and the applications that we can support. So, we're looking at end of next year, 2024, being a really high value target with a good multiple for an exit.
Fantastic, John. Listen, great. You guys have made some fantastic strides. So looking forward to hearing more. And you're going to be at dinner as well. So we'll talk to you then.
Absolutely.
Also.
Yeah. Happy to answer any questions at dinner or by email. Please contact me.